Welcome everyone. In this video today, I'm going to let you know about the cultivation of different microorganisms like E. coli, yeast, fungi. So after its isolation and cultivation, I'll let you know about gene expression process. This video will give you a basic process and protocol how to proceed and carried out experiment. So without delay, I'd like to start from E. coli. Escherichia coli. In brief, E. coli is a bacterium that can easily transform, and they have plasmid inside their cell and antibiotic resistance genes. To grow such kinds of bacteria, basically we need a media for growth like LB growth media, agar plates, sterile loop for transfer and streaking, and also what you need incubator for incubation and culture tube or flask for the uh, growth of organism in the second flask. So initial procedure of E. coli cultivation includes preparation of a sterile environment, basically for the inoculation of E. coli or even other kinds of microorganism. We need to have access of flame near to the working environment, like in a biseptic cabinet. You need to sterilize the tool before utilizing it for the scientific procedure or even a culture. Then inoculation, we will have like a single clone inoculation initially in the LB agar, and afterwards we will select the clone from the agar with the using sterile loop and transfer into the LB liquid medium for the further isolation. Inoculation, then we will do inside yeah, incubation we will do inside the incubator on maintaining the proper environment like oxygen, so temperature, even gas requirement. And bacteria will multiply within a certain period of time, and their growth we will measure with using spectrophotometer. For that we have to measure the optical density. Higher the turbidity, there will be more will be the microorganism. So after having a like a pure isolated clone culture of a microorganism like E. coli, we uh, are now ready to transform. So for the transformation, we need uh, vector. As vector, we have a selected organism. E. coli safe life is an organism, competent cell, and we are trying to integrate foreign DNA. So that will be our desired DNA for the insertion, and then we will have ice bath, recovery medium, and antibiotic are played for the selection whether the organism survives or die, provided antibiotic or not. On taking a micro organism like E. coli, we can use a plasmid DNA of E. coli to integrate into other competent cell as well. That could works. A general concept of E. coli transformation includes like what we do. We transfer on the foreign plasmid or E. coli plasmid DNA into other competent E. coli cell. Uh, so at the time we need a heat shock. So maybe before carrying out the experiment of transformation, we need to extract of DNA, and we treat DNA with a competent E. coli cell in the provided environment, and on providing heat shock, there will be a transformation of a DNA into the E. coli cell, and there will be chances of cell growth in the medium and express resistance gene. This resistance gene help to identify the organism, and another process is antibiotic selection. For the selection of antibiotic, we normally use ampicillin, kanamycin, and chloramphenicol. And those bacteria which can only survive in presence of like those antibiotics, they will be chosen as the organism resistant and able to survive in that particular environment. And so now like to let you know about the cultivation of yeast. Saccharomyces cerevisiae normally model species yeast that used to cultivate in the environment for different kinds of protein, lipid, or even a biofuel production in biotechnological industry. Normally, growth of yeast can be done in YPD medium, and then temperature for the growth of yeast inside incubator will be 30, and maintain the sterile environment on incubation carried out inside a shaking flask. Conceptual steps of yeast cultivation includes like yeast cloning from agar plate and inoculate in liquid YPD medium. Afterwards, we are having inoculation and after incubation, we'll end in the incubator and measurement of optical density for to monitor the growth of organisms. Normally, mm, yeast enzyme production uh, will be studied as well as yeah, they can be used for the biotechnological purpose. Now, I'd like to let you know about the fungal cultivation and fungal enzyme production as well. So, initially, fungal enzyme production can be carried out with isolation of a fungi. So, for the isolation of a fungi, we need a proper media. That is, normally we use a potato dextrose agar for the growth of fungi. So, sterile inoculation needle is obligatory to carry out experiment on maintaining the temperature for the growth of fungi inside incubator includes like a 25 to 30 degrees centigrade. Yeah, conceptual stage includes initially we will have like a fungal spore for the plating. Then on plating we will have a mycelial growth, and we will measure the cloning diameter over time for the estimation, and also see what kinds of enzyme they have secreted. Thank you so much for this video. I'll be back with another different video in detail in another episode. Thank you.